Great. All right. Thank you, Allison, for the introduction. And I'd also like to say a thank you to the organizers for giving me the opportunity to talk. So today I'm going to talk a little bit about CAD-assisted design and solid phase assembly of crisscross origami. So so since this is a functional nanomachines workshop, after all, I don't think I have to elaborate too much about the different techniques of achieving molecular placement and nanometer precision. But I think what is one of the exciting current challenges in this field that may not be immediately obvious is how can we scale up that nanometer resolution that we're able to achieve over like a longer range? How can we achieve both nanometer precision and long range registration? So maybe one example of how we can do this is, or something to strive for, is something like an artificial cell. So it's spanning on the order of tens or hundreds of micrometers, but it's able to harness that nanometer or atomic interaction to power the behaviors that are essential, say, for life. And so so I'm in the lab of Dr. William Shi, and since we are a DNA nanotech lab, our approach to this question, of course, involves molecular self-assembly. And so previously in the lab, we've developed a hierarchical assembly method that can produce these fully addressable DNA origami structures that are on the scale of micrometers. So you can see in the video here, these structures are themselves formed of individual DNA components. So in the red, that is a seed origami, and this is nucleating the assembly process. The bulk of the structure is made of these blue origamis, so we call them slats, and they're forming these interactions with one another. So one slat can form interactions with many others to achieve a cooperative assembly. This is an advantage for us because this helps to produce that all or nothing assembly. And so individuals, structures can assemble and achieve these metastructures that are now on the order of microns. And also, I think importantly for the application is that the whole structure is made up of these unique components and making it uniquely addressable. So some of the advantages to our assembly method that we hope will make it easier to adopt for the community is that since we're working on the level of the origami themselves, we just have to worry about the identity of the handles of those sequences. That's how the metastructure's like shape is programmed. We don't have to worry so much about the sequence design, which can be a barrier to entry for a lot of people trying to go into DNA origami. Another aspect is that because all of these slats are like structurally totally identical, so it's quite modular. So again, as I mentioned earlier, you can swap out the identity of those handles to program a different shape, or you can add different functionalizations to the structure using this. So using the Sneak, the lab has created some of the largest fully addressable DNA origamis to date, and Crisscross also has this built-in resolution of 14 nanometers. So you can see in the top left corner there. So on this origami surface, you can put any 2D pattern that you would like, and those patterns can then later be functionalized with antibodies, carbon nanotubes, so anything that you can put an oligonucleotide handle onto. In this particular example, we've used DNA again, so it's a DNA nanocube that is decorating the surface. And so I think this makes Crisscross a very versatile scaffolding for applications that might require micrometer registration. So we're in a very exciting time for Crisscross right now, so we're currently pursuing several collaborations with other institutes, exploring how Crisscross might help with research in biology, nanomaterials, and computing architecture. But there are also some wrinkles that we need to iron out in order to really accelerate the potential for application here. So broadly speaking, there's kind of two directions. So one, we want a reliable and accessible platform. So in terms of the tech, you can think of this maybe as a back end of our platform. We need a molecular self-assembly that can produce this high quality structures at scale. And then on the other hand, we really want that others can easily use this technology for their applications as well. So we really want to streamline the assembly process and make it low cost. And we also want to make it not so intimidating because there are many parts and we don't want people to have to look under the hood every time they're trying to make a design. So for the first direction, we took some inspiration from solid phase oligonucleotide synthesis, which has really revolutionized molecular biology and kind of started the genomics age as we know it, because this has enabled like a on-demand synthesis of custom DNA sequences, right? So we're trying to see how can we translate some of those ideas and then apply this to origami assembly for large micrometer structures and maybe hopefully bring about like a new age of origami assembly. So in more detail, what that solid phase oligonucleotide synthesis looks like. So it's something like this. So you start with a magnetic beads on the top left corner. So these are functionalized with oligo handles that are then complementary to the seed origami that initiates the full assembly process. So you can look at the TM micrograph there, they're very tiny. We then introduce a subset of these slat components that can then nucleate and attach to these seeds. The top right shows sort of this nucleated assembly, what it looks like. It's kind of these brushes or, kind of like whisk structure. So because of that ability to pull down and do washes very efficiently with the beads, now we can do these washing steps and we can introduce the next set of components that are required for the growth. And on the right bottom, we can see this is attachment to the beads. And then finally, when we're ready and the structure is completed, we can use strand displacement. Using DNA strand displacement, we can kick off the assembled structure back into the solution for collection. So on the left bottom, you can see on the top, that is the previous method using just assembling everything one pot in the solution versus the bottom, which is using solid phase assembly. So you can notice very clearly that in terms of the background of the excess components that were not incorporated, this could interfere with your downstream applications. So we have a much more pure and clean assembly. We can also concentrate the products as we need. And also, we're working towards this currently, but this method should help us reach even larger scales than we were able to reach before in solution. And of course, taking this analogy to phosphoramidite synthesis a little bit further, so maybe with a small alphabet, we can reuse some of these components to build any shape. An advantage of this would be that there are fewer components that we have to individually prepare in order to make a large variety of things. So here's an example of like a stepwise assembly that can construct something that if you squint maybe looks like a T-Rex, and there is the TM of that. So you can think of it kind of like an Etch-a-Sketch. You're drawing these like 90-degree patterns to produce a shape that you want. And finally, saving the best for last, so the team has produced a CAD software that simplifies this design process. The tool is open source, and it has a JavaScript front end and Python back end. And so in this example, I'm just showing how you can lay down these individual slats on different layers and have these crisscross attachments. You can also, customize the aesthetics to whatever color you would like. And key for the assembly, you need to specify where that nucleating seed is located. So we can also place a seed to a specific location. And then down here, this is a library of oligonucleotide handles that we can later use for functionalization. So we can also specify which surface of the structure to put these handles. So you can have a different set of handles that will later be functionalized with, say, something like antibodies. And then on the right, you have this fun 3D rendering, so you can see what it looks like in 3D, the structure that you have designed. So this is one option to make the structure using the software tool. You can also, once you've already made a past design, you can upload one. And so in this example, this is a larger structure with almost 200 individual unique components. So the numbers are just showing the sequence of the handles there that will cause it to assemble that way. And so this is what it looks like in 3D. And then now you can just click generate the megastructure. And this will give you a list of instructions that are ready to use to help you with the experimental side of preparing the structure. So just waits a little bit for it to generate, and you can take a look. So it spits out a CSV file that has a list of instructions for liquid handling automation. So this can translate seamlessly to our liquid handler, which helps us with pooling the components needed for making the origami components. And after those are assembled, we can then do a solid phase assembly to produce the structure in question. And we're currently also working on trying to automate that process as well to just try to make it even more streamlined. So I believe I'm out of time, but I just want to shout out a wonderful team, especially my advisor, William. And I want to especially highlight Matthew and Corey, who have been responsible for the CAD tool. So thank you for your attention.
Anyone who hasn't asked anything yet? Oh, we have two even. Okay, well, we'll try to get to both of you.
Hey, great talk, super cool results. Have you also considered making a version where you align those struts not only in a perpendicular fashion, but also get like, I don't know, triangulation, which then might affect stability and those things?
That is a great question. So because of this 90 degrees, there's no triangulation, right? So it's actually kind of floppy. You can see the angle change. So we can do 3D, we can do sort of 60 degrees and with 3 layers, so to triangulate. If you want like a very rigid pattern and you don't have any give, we can also do that more simply with like kind of cross beam. So this is a more minimal approach. But maybe if you want something super rigid, you can like stack 3 layers at the 60-degree angle. This is a good question.
Hi. So an amazing feature of DNA origami historically has been that you don't need to worry about folding pathways, right? You just mix it in a pot and things tend toward the low energy structure. So now in your solid phase assembly, I mean, you have the, if I understand correctly, right? I mean, the opportunity to design and optimize the order of adding components. And so do you want to say something about how you design, orders of addition and whether, that requires some optimization and how, you can help the user design that?
Yeah, for sure. If I try to say it maybe simply, so I think previously I mentioned this analogy of like where we have sequence design in origami, but here we're worrying about just the handle identities. So it's on an abstracted level. So we found that like we do have to be kind of careful with the handle selection because unwanted, like spurious interactions could occur as a result. So based on like, we're currently working on like algorithms and approaches to make it favorable to produce the desirable structure as opposed to like an unwanted side product. And in terms of like the stepwise assembly, so I think this animation actually shows it pretty well. So each of those steps, there's like a growth front that emerges from the seed itself because the next set of components can only attach like stably once there's enough of the like precursor, the slats that came before it. So that's, I think the route of the assembly is already a bit established, but now it's a question of like, okay, can we prevent spurious interactions? Can we avoid aggregation among the slots themselves, and then because those will not participate in the assembly reaction. Happy to talk about it more.
Thank you. We have to leave it at that, but again, there's a workshop on this afterwards. Shuntaber, you are up next.